All right, so we are on chapter 6, section 3. And last section, if you remember, we talked about globular proteins. So these are proteins that are soluble either inside or outside of the cell. And now we're going to switch to proteins that function as regulators of molecules that are passing through a membrane in the cell. So whether that's the exterior membrane or an organelle membrane. And you have to remember about your membranes that they're amphipathic, right? So part polar, part nonpolar. So our membrane proteins tend to have nonpolar amino acids that are facing out towards those hydrophobic membrane regions. And then their polar residues are facing inward into the interior of the protein. So you just kind of got to remember those key things. So that was my reminder for you. All right, so we have 3 major classes of membrane transport proteins. They can be membrane receptor proteins. So these are involved in transducing our extracellular signals across the plasma membrane, and we're really not going to talk about those until we get to chapter 8. There are membrane-bound metabolic enzymes. So these are like the ones that are in the inner mitochondrial membrane, right? They're involved in those oxidation reduction reactions and ATP synthesis. So that'll really be chapter 11 and 12. Also in the chloroplast thylakoid membrane, right? All that'll be in 11 and 12. Then membrane transport proteins, and this is what this section is all about. And these are going to be the ones that facilitate movement of either polar molecules or some other types of molecules that we'll talk about. But they're a really, really excellent example of the interdependence of protein structure and protein function. Remember how we talked about that relationship. So there are different kinds of mechanisms that allow for membrane transport, right? So we want biomolecules to cross our membrane, and they do it in one of 2 fashions. And the fashion that they move depends upon their chemical characteristic. So hydrophobic molecules, right, can diffuse. They can go across the lipid bilayer, moving from high concentration to low concentration. So an example of that would be like a steroid hormone, right? Then you can have polar molecules. Now these can't just diffuse. They actually have to be transported across the cell membrane by some kind of membrane protein, because they can't go through that nonpolar interior of the membrane. So it needs to have help. So what kind of transport proteins can we actually have? Well, the 2 major characteristics are passive membrane transport proteins or active transport proteins. Passive, they're going to facilitate movement of our molecules, but it's in the same concentration as the concentration gradient, right? Same direction as the concentration gradient. So it moves from areas of high concentration, right, to areas of low concentration. So that's diffusion. Does not require energy at all. It just happens by diffusion. The second class is active transport proteins. These are going to move our biomolecules against a concentration gradient. That means we're going to have a low concentration, and we're going to want to move it to an area of high concentration, right? This requires energy. We need energy in order to make this happen. So your book kind of goes around it, but I want to point out at this point that there's another set of classifications of membrane transport proteins. They can either be identified as carriers or they can be channels, right? And it's all about how the molecule is designed. So carrier membrane transport proteins, all of them are active transport. So all of them require energy. Carriers also tend to transport molecules very slowly, and they have very high specificity. And we'll see that. The other set, the channels. Channels can be either active or passive. And these are non-specific, and so they go very fast. They can go very fast. They go as fast as diffusion. Okay, and so what we're going to do is we're going to look at some examples of data from these different kinds of membrane transport proteins. So here are a few examples, right? So we have a couple of different kinds, right? So we have diffusion here, then we have passive transport, and then we have active transport. So the two different kinds of passive transport, right, are passive transport channel or a passive transport carrier. So what you're going to see is our passive transport channel graph looks very, very, very similar to diffusion, right? So it's all concentration dependent. The higher the concentration gradient, the faster those molecules move, right? But now, when we get to a passive transport carrier, what we can see is it's going to be linear until we get to a certain point. And then this carrier molecule, this carrier protein, excuse me, is going to get saturated. And so it's no longer going to be able to transport those proteins because they're basically saturated, right? So both passive transport with a carrier and active transport with a carrier can both become saturated. Can both become saturated. Okay, and the other two, diffusion and passive transport through a channel, are very, very, very fast. So let's talk about what makes something passive, like how is it going to actually happen? So passive transport is going to occur when you have a negative delta G, right? So what's going to determine if a molecule can cross that cell membrane? It's going to be delta G. So in passive transport, delta G is already negative. But in active transport, that delta G is going to be positive, and so it's going to require energy. Now, it's the same delta G, but we have a slightly different equation, right? R, in this case, is our gas constant, right? T is temperature, but be very, very careful. It's in Kelvin. Temperature's in Kelvin. Then Ln, C2 over C1. What is that? So C1 is our concentration. I'll just put brackets, concentration at the start. And then C2 is our concentration at the end. And we'll look at a couple of examples so you can see what that means. Z here is our electrical charge. So if you are, let's say, transporting an ion, it's going to be the ion's charge right there. That'll be Z. F is Faraday's constant. So Faraday's constant, if you don't know, is 96.5 kilojoules per volt mole. That's Faraday's constant. And then delta V is going to be our membrane potential. Membrane potential. Okay. So if necessary, if we do this calculation and we get delta G being positive, then active transport is going to couple that transport with an input of free energy to make the overall reaction of the two spontaneous. So we get that negative delta G. So I want to do an example, but I don't really have room. So I'm going to just stick in an extra page. Maybe. Ha ha ha, all right. All right, so we'll take an example of the sugar transporter. Whoa. In the sugar transporter, usually when sugar molecules are transported in, you have 100 times the amount of sugar extracellularly, EXTRA, extracellularly. I think my brain goes too fast and I leave out letters. I think that's a problem. As compared to our intracellular. So not equals, but as compared to 1x intracellularly. So remember, when we have a ratio, right? If we go over here, where did I add it in? I added it in a weird place. That's okay. Remember, when we have a ratio, it doesn't actually matter what our units are for here. Just like we talked about in the earlier chapters. So it doesn't really matter as long as your units cancel out. Okay, so you have way more sugar extracellularly, so outside than inside. Sugar, can you tell me about the electrical charge of sugar? The charge on sugar is 0, right? Temperature, let's see, we know body temperature is 37 degrees, but that's Celsius, but we know our temperature has to be in Kelvin. So convert that to Kelvin, you should get 310, right? So if we plug this in, you're also gonna have to know, no, you don't, right? Because our charge is 0, so our unit of Z and our Faraday's constant times our volt, this whole unit is gonna be 0 because our charge is 0. So I'll show you. Okay, let's plug it in. Let's plug all this information in, okay? So we have delta G is equal to RT ln concentration 2 over concentration 1 plus 2 times Faraday's constant times membrane potential. Okay, so delta G is equal to 8.314 joules per mole K. But what did we say where was that? I know I'm gonna have to flip back and forth, right? Here, that our Faraday's constant is in kilojoules, right? And our gas constant needs to be in kilojoules. So when you put it in, make sure you put it in as kilojoules. So that would be like 10 to the negative 3 kilojoules per mole Kelvin, right? Then we put in our temperature, which was 310 Kelvin. Then our concentration, I think I'm so gonna run out of room. How do I always run out of room? Okay, times our concentration, so that's ln of concentration 2 over concentration 1. Concentration 2 is where we're trying to go. So that's the inside of the cell. So we said that was 1, whereas our extracellular was 200, right? Now, if our Z is 0, 0 times any of this stuff, right, is gonna be 0. So we don't even have to worry about that. So what you should get for a delta G, right, is something that is negative 12.0, check my math, kilojoules per mole, right? So that means that you're gonna have passive transport, right? This delta G is negative. It's gonna be spontaneous. So you're not gonna have to couple it, right? So let's do another example. Let's do Let's do the movement of calcium 2+, right? And this ion transport is super important in muscle cells, right? So if you have calcium ions in the cytosol of a muscle, right? What you want to do is move that calcium from the cytosol to the inside membrane, to the inside of the membrane-bound organelle called the sarcoplasmic reticulum. And we'll talk more about this later, but it's super critical for muscle contraction for the calcium ion to go from the cytosol inside the sarcoplasmic reticulum, okay? So normal calcium levels in the cytosol are about 0.1 micromolar. Ooh. Calcium concentration in the sarcoplasm is 1.5 millimolar, right? So we know these are two concentrations. Let's get them in the same units. So that'd be 1,500 micromolar, right? In the sarcoplasmic, not sarcoplasma, sarcoplasm, plasmic reticulum. reticulum, reticulum, I do believe. All right, so that's an uphill gradient, right? We're trying to move against a gradient here. That's a massive gradient. That's like a 15,000x gradient, right? So I'm thinking we're gonna need energy for this. So what's Z for our equation? It's gonna be +2, right? Remember Faraday's is 96.5 kilojoules per volt mole. Doesn't matter which one you put first, volt mole. Our temperature is again going to be body temperature, so at 37 or 310 Kelvin. And then we also need to know our membrane potential. Our membrane potential I will give you, and I'll tell you it's 50 millivolts, but look over here, we're in volts, so we need to convert that to volts, okay? All right, so now we're going to plug it into our equation. Delta G is equal to RT ln C2 over C1 plus Z, it's our charge, Faraday's, times our membrane potential. Okay, so plug them in. 8.314 times 10 to the minus 3, remember, because we want to be in kilojoules, mole K plus, not plus, times natural log of, now, remember this is C2, so where we're going has a concentration of 1500 micromolar. Where we're starting is 0.1 micromolar, right? That's crazy. So plus 2 is our charge. Faraday's is 96.5 kilojoules per mole per volt, right? And then we have our membrane potential that I can't quite fit in, but then that would be again times 0.50 volts, right? Okay, so when you do the math, what do you get for delta G? You should get delta G 34.7 kilojoules per mole. So is this going to happen spontaneously? No, it's super unfavorable, right? So the cell is going to use the energy equivalent of ATP hydrolysis. It's not the hydrolysis of the bond, it's not the breaking of the bond that has the energy, but it's the coupled reaction that provides that energy. And so you get about 50 kilojoules per mole of ATP, right? And so we'll learn about that in a little bit. Okay, so those are our two examples for how we can calculate membrane, whether it's going to be spontaneous, whether it's going to just diffuse across the membrane, or we're going to have to actively transport it. Okay, so let's look at some different structures, right? So the typical structure, one of the typical structures is a beta barrel. So porins are typically beta barrels, and a beta barrel is just a structural motif. And so in this kind of a motif, right, our primary sequence, so the list of amino acids contains alternating polar and nonpolar residues. And why is that, right? Because if you have a beta sheet, the amino acids, as they go sequentially, right, they're going to have side chains. One's going to stick out and the next one's going to stick the other way, right? And so they're going to alternate. And so you got to think it'll be polar, nonpolar, polar, nonpolar. So all the polar residues are on one side, all the polars on one side, all the nonpolars on another side, right? So that our nonpolar residues are going to face that outward towards the membrane, right? And our hydrophilic charged are going to face on the inside of the pore. So they can interact with whatever polar molecules are being transported, right? So you can either have really, really selective or really non-selective porins, right? And it's going to depend on the number of beta strands, which determines the porin interior diameter, right? So if you have more beta sheets, your barrel is going to get bigger and bigger and bigger. If you have fewer beta sheets, it's going to be a smaller barrel, right? And so then it's also going to depend on, those side chains that are sticking inside the barrel. So the chemical properties of the amino acid side chains that line that interior channel are going to tell how much specificity or selectivity is inside of those porins, right? So here's kind of an example, right? So we have a porin that's a passive transport protein. And so this is this little diagram, and they don't say this very well, but this is in gram-negative bacteria where you have an outer membrane and you have an inner membrane. And between the two, you have this periplasmic space. And so that's that compartment just located between the inner and outer membranes, right? And so you have a passive porin transport protein, right? This is this orange one. And you can have these ions or molecules that can just pass through, right? And so the reason they can pass through is because maybe they're chlorine ions, right? They're negatively charged, right? So if they have negative charges, then guess what's going to be pointing towards the inside of our porin? Well, we're going to have positively charged amino acid residues like arginine that are going to be on the inside of the channel. So they're going to be attracted to one another, and those negatively charged ions are going to be able to go through. Malate is an organic acid that has two negatively charged carbonyl groups, and so those will actually be negative at physiological pH. And so It doesn't have to be an ion, but it has to be something small and charged, right? Okay, and then we'll talk about how that's coupled to some other transport molecules. But the one I want you to focus on in this picture is this one right here, right? Okay, so what's sort of interesting is that we would think a barrel shape is really great for transport of proteins, and it is, but it's not the most common shape for a membrane transport protein. The most common shape is actually several alpha helices, which, like, it's a helix, and then another helix, and then another helix, and then another helix, and it'll form kind of a channel. So all the helices will fit together and form a channel. So usually these are very, very selective transmembrane channels. A good example is the potassium ion channel. So the potassium ion channel, oh, and remember that these are passive, right? These are passive, okay? And these occur, the potassium channel occurs in both bacteria and eukaryotes. So you would think that, right, if it's a channel, if it can transport potassium, well, could it transport something like sodium as well? Well, it's actually 10,000 to 1 selects for potassium over sodium. But potassium and sodium are super similar, super similar. So why? How can it be so selective? Well, you know, you need some kind of external stimuli that's going to trigger the activation of these channels. And we're not going to talk about that yet, but it's going to open the channel. So here, the channel will open, and both potassium and sodium will enter the chamber on the cytoplasmic side. So you have both entering the chamber. But what we see come out of the bottom of the chamber is only our potassium ions. And sodium and potassium ions, ionic radius is super similar. So they only differ by like 0.4 angstroms. So how is it that the pore can differentiate between the two? Well, what's interesting is that potassium needs 8 waters per ion in order to be soluble, right? And if you look on the inside of this pore, what you're going to notice is that you have 8 amino acids with carbonyl oxygens that can interact with potassium. Perfect, exact ratio, right? Well, sodium, on the other hand, needs 6 waters in order to be hydrated. So the amino acid side chains don't line up perfectly. They're not appropriately spaced for potassium. So it doesn't interact with potassium very well, and potassium kind of gets kicked back out. So sodium gets kicked out, sodium gets kicked out, and potassium can come through because it matches the number of charged residues on the inside matches the number of waters that potassium needs. So I'm gonna show you at the end some animations, but I gotta do it in a different video because I'm having trouble with that, but I'll show you at the end. Okay, so what's really important to take away from all of this stuff is that there are really small details in our protein structure that can lead to super important characteristics of our protein's function. So super small details, but they're very, very, very important. Okay, so let's talk about a different kind of passive membrane transport protein. These are called aquaporins. And you see aquaporins in red blood cells, in sweat glands, in kidney cells, right? All areas where we're dealing with a lot of fluid, a lot of water. And their job is to transport our water molecules across our hydrophobic membrane. But they're not super, super specific because they can also transport urea, they can transport glycerol, a few things, right? But humans have 11 different aquaporin genes. And these genes are very, very, very highly conserved. And they code for proteins with 6 transmembrane alpha helices. So if you have 6 transmembrane alpha helices, right, and that's going to make one basically pore, then this one pore is going to associate with 3 other 6 membrane alpha helices, right, to form a protein complex. So it'll basically be a 4 subunit protein complex. So each subunit has a channel that allows water to pass through. And so one of these tetrameric protein complexes can transport, get this, 3 billion water molecules per second. 3 billion per second. Oh my gosh. But that is dependent upon a concentration gradient, right? So, but that's still amazing because the pore size is so small that only one molecule of water can fit through at a time. But it can still, it's got 4 channels, but still only one water molecule at a time, and it can transport 3 billion water molecules a second. That's kind of amazing. All right, so let's talk about active membrane transport proteins. So here's our kind of jumping to our other type, right? These require energy to pump molecules across the membrane, right? So here you're going against a gradient. So there are 2 classes. There are primary active membrane transport proteins, and then there are secondary active membrane transport proteins. The primary ones, they use energy conversion processes like ATP hydrolysis to pump our molecules across the membrane. Secondary depend on stored potential energy from a concentration gradient that's already there in order to move a molecule. And so we're going to talk about each one, and we're going to start with primary, and then we'll understand secondary. You got to kind of get primary before you can get secondary. All right, so right here are our 3 kind of examples of active membrane transport. We have our primary active transporter, right, where we're seeing that we have to have ATP in order to move. We're moving from areas of low concentration to areas of high concentration, right? So our ATP turnover is going to drive this movement up the concentration gradient. So this is a primary active transporter. Now, there are 2 kinds of secondary active transporters. You can have a secondary antiporter or a secondary symporter. So let's talk about the symporter first. In a symporter, right, we have a high concentration of these red molecules. Well, that's great, but we really want to get these blue molecules into the cell. So what's going to happen is a red and a blue are both going to go in, and the red goes in because of the concentration gradient, and the blue goes in because it's coupled, and then they both get spit out. Ooh. Then they both get spit out, right? The red's going with its concentration gradient, and the blue is going against. So, but the 2 are transporting in the same direction, so that's why we call this a symporter, right? That's the symporter. Now, in antiporter, we have these red molecules that are super high concentration, right? And so these are going to go along their concentration gradient, so from high concentration, right, to low concentration. But when one of those red molecules gets transported, going in the opposite direction, one of these green molecules is going to get transported. And this green is going against its concentration gradient. So we have a low concentration here, we have a high concentration here. So we call these antiporters because the molecules are going in opposite directions. We have our one that's a high concentration gradient going this way, say into the cell, and then the green one is going out of the cell. So this is antiporters, this is symporters. Okay, so let's talk in more detail about primary active transporters. There are 2 abundant types, and we're going to talk about each one. One is the P type, which is the phosphorylated type, and then the second one is an ABC transporter. ABC stands for ATP. They both use ATP hydrolysis to drive super big conformational changes in our protein complex. And these are really very important, and these are also targets of drug therapies. There are drugs that inhibit these kinds of transporters that we can use. One example is Prilosec. That's going to inhibit the transporter that pumps hydrogen ions into the stomach. And so it's actually a hydrogen ion and potassium ion ATPase. But you know, you can have stomach problems when you have too much hydrogen ions in your stomach, and so we can target that and we can inhibit it. Zoloft is another one. It's a serotonin transporter. It targets a serotonin transporter. So normally when your neurotransmitters fire and they release those neurotransmitters like serotonin, the serotonin molecules get recycled very quickly. And sometimes they get recycled so quickly that they don't basically have a chance to take effect. And so Zoloft will actually inhibit the serotonin transporter. So the cells take longer to recycle the serotonin. And so it's like you keep getting the signal that you're getting serotonin. And serotonin is one of those neurotransmitters that make you feel happy, right? And so this is an antidepressant medication. Cocaine will actually do the same thing, but instead it'll inhibit the dopamine transporter. And so when you take cocaine, you get the feeling of like euphoria, right? And so there's nothing in life that can compete with the level of euphoria or the level of dopamine that you get when you take cocaine. And that's why people get addicted to it and have serious, serious problems. All right. That was a side trip. Sorry about that. All right. So let's talk about P-type transporters. So we said P-type, P stands for phosphorylation, and this phosphorylation drives conformational changes in our molecules. So an example is the sodium potassium ATPase. This is an integral membrane protein, so it integrates into the membrane. And it exports, so it sends out 3 sodium ions out of the cell for every 2 sodium ions that are imported into the cell. So this is really what is responsible for maintaining the electrochemical gradient. It's so funny because I can't speak and spell at the same time. It's so sad. Across most animal cell membranes, almost all animal cell membranes, right? So let's see how it works. So here's our sodium potassium ATPase, right? So what's going to happen is that we have we want to keep a charge difference, right? And so how do we do that? Well, here we're going to pump 2 potassium ions. At the same time, we're pumping 3 sodium ions. The sodium ions are going to exit the cell. The potassium ions are going to enter the cell. So how does this transport actually happen? Well, the pores in one particular conformation, right? And so the molecules can bind on either side, the potassium and sodium. And then what happens is the cleavage of ATP into ADP plus PI is actually going to induce a massive conformational change that moves the potassium into the cell and it moves the sodium out of the cell. So it's this massive conformational change that facilitates the movement of the ions. So the big thing is you have a conformational change that's dependent on ATP, right? So what's your end result? Your end result is here you have very high concentration of sodium, right? And then in our cytosol, we have a high concentration of potassium, but it's not equal. So we have a difference in charge. And so if you compare the total charges outside to the total charges inside, there's less inside, and that's why we express it as negative. So we have that charge difference, and that charge difference is called the membrane potential. And so you end up with a membrane potential of about 70 millivolts, right? And that's really, really important for neuronal cell function, right? Because you have to propagate an electrical charge along a nerve cell. And it's important for when you get to the end of the nerve cell and you get to the axon and you need to fire from the axon from one cell to another cell. So how do you discharge those neurotransmitters? That's axon firing, and that depends on your membrane potential. So it's super, super important. It's so important that about 25% of the ATP that's hydrolyzed every day by animals that are at rest, not ones that are moving, but ones that are at rest, are used to just maintain the sodium and potassium electrochemical gradient. 25%, that's a lot. That's a lot. All right, here's another example of a P-type transporter. This is called the Circa So it's the sarcoplasmic, no, it's the SERCA, S-E-R-C-A. So it's the sarcoplasmic endoplasmic reticulum calcium ATPase. If you can remember that one. All right, so what you got to remember, and I don't like the way your book did this diagram, is that this protein is sitting inside of our membrane, right? Here's our membrane. Just going to kind of draw it out because it's really important to remember it's sitting inside of a membrane. And I'm going to go ahead and add it across just so that we can kind of remember what's in the cytosol and what's in the sarcoplasmic reticulum lumen, L-U-M-E-N, lumen. Okay, so what happens is inside the lumen, you have hydrogen ions. Those hydrogen ions are going to bind down here in this part of the molecule, right? And so ATP is going to bind up here at the top in the cytosolic area. So when ATP is hydrolyzed, two things are going to happen. The first thing is that our hydrogen ions are going to exit and they're going to go into the cytosol. So our hydrogen ions are going to go into the cytosol. And then at the same time, we have calcium ions, so Ca2+, that are going to actually enter our transport protein. Okay, so we basically switched hydrogen ions for calcium. So we exchanged H+ for Ca+, right? And so at that point, right, your ADP, because we hydrolyzed it, so now we have ADP and PI. So this phosphate group there phosphorylates this little portion of our transporter, right? It's dependent upon phosphorylation. So there's our phosphorylation event, and then our ADP leaves, right? So we see that this pocket no longer has ADP in it. So the ADP leaves, and this releases our calcium from the transport protein into the lumen, right? And then what happens is ATP is a new ATP molecule is going to bind, which is going to allow this molecule to pick up another 2 hydrogen ions, and then the cycle goes again and again and again, right? So that's just our extra example. All right, so now we're going to talk about the second type of primary active. So this is second type of primary active transport molecules. So these are ABC transporters. So these are also ATP dependent, right? Because it's a primary active. And it can either import or export depending on which molecule it is, right? And so it's, again, that ATP hydrolysis that induces a large conformational change. And in this case, now you're going to change the, basically the inner pocket from one time facing outward. So maybe it's facing the cytosol, and then it's going to hydrolyze ATP, and now that pocket is going to be facing the cytosol of the cell, right? So the exterior of the cell, the cytosol of the cell. One example is a multi-drug resistance protein pump, and it removes toxic compounds from our cells. So this one is actually sitting like this. It's sitting facing the cytosol. It'll pick up some kind of toxic thing and dump it out extracellularly, right? And usually this is a really good thing. Yay, we want to get rid of toxic things that are in our cells, but it's a problem when we're trying to treat cancer patients, right? Because cancer cells can actually, excuse me, become resistant to chemotherapeutic drugs by increasing the number of these transport proteins on the cell membrane. And so they actually pump, you treat the person, the cancer cells are going to pump it out before it can kill the cancer cells, and then the chemotherapeutic is then going to have way more off-target effects, and the person's going to get sicker and sicker, and they're not going to be treated. And so it can be a real problem. All right, so how do they work? It's really a 3-step model, right? So in the first one, we have binding of our substrate to a substrate carrier molecule. So that's our little blue little C right there, right? So the substrate is going to get stuck inside of the substrate carrier protein. the carrier protein is going to bind to the transmembrane domain, right? So here's the transmembrane domain right on the periplasm side, okay? So like it's not really the outside because this is bacteria, but it's that space between the two membranes. And it's going to do two things. It's going to induce a conformational change that is going to expose the internal substrate pocket. So here's the internal substrate pocket to that substrate. So the substrate is going to be able to go in. Now the other thing that happens, you got to look at the bottom. You see here, this is the nucleotide binding domain. And initially, they're not aligned very well. And when the substrate carrier protein binds with a substrate with it, those nucleotide binding domains are going to come together and they're going to form two ATP binding sites. So now you can see the binding site is aligned properly and we've got two ATP molecules inside of it, right? All right, now the next step is dependent upon ATP hydrolysis. So after ATP is hydrolyzed, it's going to open up our substrate binding pocket. This time the binding pocket, right, is going to be exposed to the cytosol. And so the stuff that we're trying to transport gets ejected into the cell, into the cytosol. And then what happens there afterwards is our ADP and our PI get kicked out and they get replaced by new, fresh ATP molecules, and we revert back to our resting state. And so that cycle will continue and continue and continue, right? So in all of our ABC transporter proteins, we need both substrate binding and we need ATP hydrolysis to induce our conformational change. So you need substrate binding and you need ATP hydrolysis for conformational changes, right? So what's cool about this is that you need to be able to close off your binding pocket from the periplasmic space, right? If this is your binding pocket, you're going to start off like this so that you can pick up whatever substrate you're trying to transport. But remember, you're going against a concentration gradient. So if you just opened up this way, right, you're going against the gradient. It's just going to want to go back out. So what happens then, instead of just opening it up all the way, is that you close the top and open the bottom. And it works because the substrate binding site is only accessible to one side or the other at any given time. So your book equates it to an airlock inside of a spaceship, right? So your astronaut goes into the airlock, right? And the airlock has air and all that kind of stuff in it. And they close the door to the rest of the spaceship. And then they open the airlock door and then they can exit the spaceship because it's now depressurized when they open that airlock. Okay. All right. So, I did want to tell you another example of an ABC transporter protein is the cystic fibrosis transport protein. You know, in cystic fibrosis, you have to have transport of chlorine ions across the membrane, and that helps to control our movement of water across the cell membrane. And so in patients who have cystic fibrosis, they make very, very thick mucus because they don't have enough water leaving the cells. And so they tend to get infections and stuff in the lungs because they can't clear that mucus. So another example of an ABC transporter is the cystic fibrosis transporter. Okay, now we're going to talk about secondary active transporters, right? So they're going to use the energy that's already available from downhill electrochemical gradients. So think about this, right? This is a symporter. This is, you just have to kind of think about it that way, right? So let's say you have a membrane and you have hydrogen ions and you're going to use ATP to transport your hydrogen ions across the membrane, right? So this is an ATP dependent. This is like the electron transport chain, right? So you're using, in order to make our secondary active transporters go, we're going to use energy from a steep proton gradient. So what we get here is a proton gradient. To create that proton gradient, we had to use ATP. But the secondary active transporters aren't going to use ATP. They're going to say, okay, there's already a gradient here. I'm going to hijack that gradient. So our example is our lactose permease transporter, right? So these little white molecules are hydrogen ions. What you can see is there are already a lot of hydrogen ions on the top side of that membrane, right? So the ATP dependent activity has already happened. Now we're just going to take advantage of that. And I want to move lactose into my cell. So if I can co-transport the hydrogen ions and the lactose at the same time, now I can move in what I want by hijacking the proton gradient, right? And so it works in a very similar way. You have an opening at the top, right? Then they get locked inside of the inner space. So this is our first conformational change. So you have to have two conformational changes to make this go. The first one is the closing of the pocket. And then the second one is you need to open it. You're going to come back, and then you're going to go back to that outward conformation, right? So it's really not, this one actually is really not well understood how our conformational changes are induced. We really think it involves protonation and deprotonation of the amino acid side chains inside of that substrate binding site. But, you know, we're still working on it. All right, another example is the sodium iodine symporter. This is in your thyroid. And in your thyroid, you need iodine in order to make thyroid hormones like thyroxine and triiodothyronine to regulate our metabolism. So how are we going to do that? Well, we're going to need two things. I want you to kind of keep these separate, right? The first thing that we need is a primary active transporter to make a gradient. So how are we going to do that? We're going to utilize ATP. We've got ATP here, right? And we're going to be able to bring in potassium ions, and we're going to kick out sodium ions, right? So now we have a high concentration of sodium ions outside of the cell. Well, now I can use a symporter, and this is my secondary active transporter. And I can co-transport sodium ions and iodine ions into the cell, right? So my primary active transporter was an antiporter, and my secondary active transporter is a symporter. And I linked the two. So my secondary active did not use ATP, but in order to make the secondary active transporter go, I had to have a primary active transporter that used ATP. So they're dependent upon one another. All right, so that is the end of our transporters. I'm going to make a separate little video that has the animations from this section. Have a great day, y'all.